Okay, great. Can everybody hear me? Awesome. I'll just echo what others have said. It's great to be here presenting in person. Thanks for the organizers for inviting me and letting me tell you a little bit about some of the work in our lab. So we use human-induced pluripotent stem cells, and right now our lab is really focused on using these models to understand cellular mechanisms that would drive Alzheimer's disease pathogenesis and how this may also be related to understanding more about brain and neuron aging. Oh, sorry. So there are a few little weirdnesses in this because of the transfer. But so I thought what I would do is just start with thinking about what is neurodegeneration, the progressive degeneration of the structure and the function, which I think is really important, of the nervous system. So I've listed a couple of common neurodegenerative diseases that we've all heard about. And of course, as you have heard multiple times today, that aging is the most common risk factor for nearly all of these. And what strikes me is that if we list sort of the clinical and pathologic hallmarks of these diseases, what really is, I think, prevalent in each one at the cellular and molecular level is the loss of cells, particularly neurons, and proteinopathy, the accumulation of abnormally folded proteins. And so my lab is focused on Alzheimer's disease, and this is, of course, apparent in AD, where you have massive loss of multiple regions of the brain, primarily due to neuronal loss, due to the loss of the functional unit of the neuron, which is the synapse. And I'll get to that sort of at the end of the talk. And then, of course, we have the common pathological hallmarks, the amyloid beta and tau pathology seen in the brain. And has been reiterated several times, I think pretty much on all of the slides I've seen today that have talked about how with aging increases disease is Alzheimer's disease. So we know it's an aging disease. We know that it almost doubles in incidence as people age, and that there's very few incidences of Alzheimer's disease in people under the age of 65. And this is really important because we not only have an increasing aging population, at this point, we don't have a good lead on any kind of therapy that would actually reverse or halt the symptoms of AD for a reasonable period of time. And to me, this has always been a challenge, and I think you can apply this to many complex organs in the human, but why we've struggled so hard with AD is I think it's been very difficult to understand an accurate picture of what's going wrong in cells. So obviously we know the human brain is hard to access, and in all of these neurodegenerative disorders, they're fairly long and they're progressive. And I will argue that I think we don't even actually know when the symptoms are starting for some of these. And so how can we prevent something when we maybe have already gone too far? And of course, in humans, we have a very complex interplay of genetic and environmental factors. So the approach I've taken now for the past 10 years or so is to try to use pluripotent stem cell technology to understand this. So you're all familiar with this, but we can take a somatic cell, we can reprogram it, and the power here is we can now differentiate any cell type of the body from these cells. So we actually have the ability now to have living human neural cells and understand the cell biology that's going on in isolation, purified cell types, and then putting them together with other brain cell types, and as Kira showed, even cell types that are outside of the brain. Now, there are caveats like any model system. I'm not going to go into this very much in this talk, but happy to have a discussion over coffee that, this technology does reprogram cells. It does kind of rejuvenate them to an embryonic-like state. They have loss of certain key markers of aging. But despite this, for the last decade, numerous studies have been able to report relevant AD phenotypes in these iPS-derived cells. And there's a lot of work going on how to really incorporate cellular aging into this model. And, I have some ideas on that. So when we think about this, we want to use this model to think about this at the cellular level. And so what I like to do is think about how the hallmarks of aging, but then also on a cellular level. And so these are intricately associated together, what goes wrong in cells and aging and also in Alzheimer's disease. So what I did here is I just did a really brief PubMed search where I typed in every hallmark of aging and Alzheimer's disease. So you can see that each of these hallmarks of aging has an associated pathogenic event that's been that occurs with Alzheimer's disease. So what I want to do is highlight two recent projects from my lab. And I'm going to do this at a very high level that are starting to try to understand in our neurons what might be going on at the cellular level. So the first I'll talk about is a study we have on looking at dysfunctional endocytosis and vesicular trafficking, and that could be contributing to proteostasis. And that's really been led by an M3D student, Allison Knupp, and a postdoc, Swati Mishra. And then finally, a project on looking at altered expression of genes that regulate epigenetic factors and how that might lead to mitochondrial dysfunction. And this study is really spearheaded by Fred Yaboa, who is an MCB student, and Harry Frankowski, a research scientist. So let's talk about part one. So we're really interested in how trafficking of proteins through the endolysosomal network. So this is really important in all animals and all cell types. It regulates the intracellular location of proteins, and it's essential for homeostasis, function, and physiology. And in terms of AD, it's really becoming a very interesting interplay of genetics and cell biology. And I'm sorry again for the slide, but recent genetic studies have found about 12 to 15 genes or even more that are associated genome-wide with increased Alzheimer's disease risk that function in the lysosomal trafficking. And we think that it's a really good way to apply this genetics to the cell biology, because if you look at the main cell types of the central nervous system, including neurons and glia, endocytosis is critical for their function. And all of these genes that are associated with increased AD risk may play different roles, and this may be very unique to each cell type. And for decades, this phenomenon of enlarged endosomes, which are indicative of dysfunction, have been observed very early in AD pathogenesis, prior to accumulations of the pathogenic hallmarks of amyloid and tau. So there's something about protein trafficking that we think may be a really early driver of the disease. So we have this hypothesis that loss of function of these endosomal genes will yield measurable and hopefully modifiable phenotypes that would be relevant to neurodegeneration in our iPS-derived cells. So the story I'm going to tell you today, we looked at one gene called SORL1 or SORLA, and we used a CRISPR approach to knock it out in our iPSCs and also introduce AD-associated coding variants on one allele and use these cells to define cell type specific phenotypes. And we can differentiate both neurons and glial cells and test. We want to use this platform also to test small molecules that modify these phenotypes. And in the interest of time, I'm only going to tell you about our neuron data. So SORLA, or the SORL1 gene, the SORLA protein, has been associated with increased AD risk for years, and the biology is quite relevant. So SORLA is an endosomal sorting receptor, and it functions at the early endosome. So as you may remember from cell biology, cargo gets internalized in the cells. It hits this hub of the early endosome, and it can be sorted for degradation. It can be sorted via another protein complex called retromer back to the Golgi, and it also can be sorted outside of the cell in a recycling pathway. And the canonical cargo of SORLA is, of course, APP, the amyloid precursor protein. So a lot of work has been done about how this pathway regulates amyloid beta. But I will show you that we think it's important for other neuronal cargo as well. So the first thing we did is we knocked out SORLA in our iPS neurons. And here you can see we have loss of the protein. And we saw a phenocopy of what I showed you in the beginning that had been seen very early on in AD brains, of these presence of these enlarged endosomal structures. And what it turns out is that these enlarged endosomes are really leading to traffic jams. So they're trapping cargo in the early endosomes. So we have, sorry, I put the wrong graph in, but this is the APP, and actually what we see is there's more APP in early endosomes, and also with TrkB. And the important cargo I want to talk to you about here is the subunit of the AMPA receptor called GluA1. And the AMPA receptor is really important in neurons for making functional synapses. And so actually all of these proteins, although this is the wrong graph depicted here, are stuck in the early endosome, and this impacts how they're trafficked to other compartments. So we can show in data I don't have time to talk about impacting of how these cargo go to the lysosomes. But what I really wanted to show you is that it really is important for how these proteins are recycled to the cell surface. And that this is a key event that could impair neuronal function. So in order for functional synapse, these receptors go through the recycling pathway through recycling endosomes and are delivered to the cell surface. And what we found is that in our neurons where we've lost this sorting receptor, we have a decreased number of these subunits on the cell surface. And in very preliminary data, we think this may actually impact the neuronal function. So if we put these neurons on a little chip called a multi-electrode array where you can actually measure neuronal firing, we see that over time, our knockout cell lines are firing less than their wild type. So the next question we wanted to ask is really whether this is a modifiable pathway. And in itself, this molecule is not an attractive drug target. It's large, it's not easy to activate or inhibit it, but we think that related proteins in the pathway could be better. So we've been working with a molecule called TPT260, and this molecule enhances the function of a multi-protein sorting complex called retromer, which I showed you a couple of slides earlier. And in this experiment, what we did is we were showing a few different things. We have our sorla knockout neurons with enlarged endosomes, and then we made an allelic series where we have loss of either one copy or we've introduced AD-associated variants onto one copy of the sorla alleles. And all of these neurons show enlarged endosomes. And interestingly, when we treat with this small molecule, we can rescue the phenotype in all of our cell lines. And also what's interesting is that in wild type cell lines, this molecule has no effect, suggesting that there is a limit on the size of these vesicles. So in summary, for this part, we can use this model to recapitulate a very early AD cytopathology. We can show that this leads to traffic jams, that it impairs trafficking of key neuronal proteins. And hopefully we can use this model to test novel molecules as proof of concept for therapeutics. And in the future, what we want to do is generate cell lines from patients who have multiple variants in these endolysosomal genes and investigate polygenic risk. And we want to further define the molecular phenotypes by performing transcriptome and proteomic analysis in these cells. So briefly, I just want to take you through the second part of a story in the lab where we're using this model to look at what might be going wrong in the cells in AD. And that is looking at abnormal expression of epigenetic regulators and how it might lead to mitochondrial dysfunction. So in this case, our hypothesis is that epigenetic dysregulation is apparent both in aging and in AD. And we think that altered expression of these regulators in iPS-derived neurons will impact both aging and AD cellular phenotypes. And we focused on a gene called HDAC2, or histone deacetylase 2. So this is a deacetylase. It removes the acetyl groups from histones, thereby compacting the chromatin and causing repression of gene transcription. And in animals, this has been shown to impact synaptic function in memory. It's upregulated in AD, and it's also upregulated in aging. So there's a thought that this may be contributing to some of the cognitive decline in both of these conditions. And when I started here about 5.5 years ago, I entered a collaboration with Rick Morrison in neurological surgery, who unfortunately and very untimely passed away in 2019. But his group had worked for decades on the mechanisms of neuroprotection in a lot of different neurodegenerative models. And what they had shown was that really for one of the first times is that this HDAC, in addition to regulating synaptic function, et cetera, regulated expression of mitochondrial genes, in particular mitochondrial genes that were had specific isoforms in neurons. And one of these genes is called endophyllin B1C, and it's multifunctional mitochondrial associated protein. And what they were able to show is that it's protected against a cell death in primary neurons and also in patient brains from our own Alzheimer's disease center here, it's lost. And so what Harry and Fred did was just start to look at this in our iPS-derived neurons. So we take our stem cells, we can make a progenitor population and differentiate and mature these cells over the course of several weeks. And what we noticed is that just endogenously expression of HDAC2 went down, and this correlated with this neuronal specific isoform of endophyllin B1, which is shown as the upper band here. At the protein level, these two isoforms migrate at the same molecular weight, but at the RNA level, we can clearly document increases in both of these RNA isoforms. So we wanted to ask, or we started thinking, is really downregulation of this gene, is it enhancing pathways that are impacting neuronal maturation, potentially neuronal function, and would that be important for looking at protection from neurodegeneration? So very quickly, I'll just show you, we did a couple of experiments looking at how downregulation of HDAC2 influences mitochondria. So we can use an shRNA to downregulate HDAC2. We see, interestingly, upregulation of ENDO-B1C and downregulation of the mitochondrial protein MFF, which promotes fission. So subsequently, we could see elongated mitochondria. We could also see elongated mitochondria when we just overexpressed this downstream gene, endophyllin B1C, suggesting that there was a confirming really the link between HDAC2 regulation of endophyllin B1. What then became interesting was we wanted to look at the functional effects of the mitochondria. So we asked how it influenced mitochondrial respiration. And so we performed seahorse assays. And here we could see that when we knocked down HDAC2, we saw an improvement actually in all functions of the mitochondrial, the basal level and the respiratory level, while there was no effect by overexpressing endophyllin B1C. So this also shows that in addition to endo-B1C dependent pathways that may function at elongating the mitochondria, HDAC2 is doing more to activate genes that may be affecting mitochondrial respiration. Finally, we were able to show that we could recapitulate previous data showing that this increases synaptic gene expression, and also if we knock down HDAC2 or overexpress this endo-B1C, we can protect against a neuronal cytotoxic insult. And finally, we can look in wild type and also neurons that are derived from Alzheimer's patients, in this case, patients with a duplication of APP that develop early onset Alzheimer's disease, and show that knocking down of HDAC2 reduces levels of amyloid beta. And in very new data that we think could be exciting, we can use a small molecule that inhibits HDAC2 and see a very similar effect. And this is promising because several HDAC inhibitors are actually already FDA approved molecules. So for this part, we can show that decreasing HDAC2 improves these mitochondrial dynamics and function, that it's neuroprotective, it enhances synaptic gene expression and reduces A beta. And for our future work, we're really interested in looking at more global pathways. When we knock down the cells both at the level of the transcriptome and the metabolome, given its effect on mitochondrial respiration, and exploring how these mechanisms may protect against age-related neurodegeneration. So just to summarize, we really feel that our IPS model can provide mechanistic insights into age-related neurodegenerative disease by providing biologically relevant and living cells to dissect these mechanisms. We can provide functional validation of genetic or pathologic studies. And as Kira showed you a little bit, we can move towards three-dimensional modeling and putting more of the relevant cell types in our models to enhance the complexity and the accuracy of these systems. So thank you for that, sticking with me through all of that. And I just want to acknowledge my lab and our funding. So I'm happy to take questions.
Hey, that was really interesting. So regarding the enlarged endosomes that you see, do you also see differences in pH of those endosomes? And do you see like a failure of the endosomes to mature from a Rab5 to a Rab7
That's
positive endosome?
That's a great question. So we haven't directly measured pH, but what we see in data I didn't show you is a failure of substrates to either get to the lysosome or be properly degraded at the lysosome. So we need to do pH measurements there. We have not seen a difference we haven't been able to really track individual endosomes to know if the ones that are more enlarged are not maturing faster. That's a great idea and suggestion, but we don't notice a difference in the number of early versus late endosomes that we see.
Yeah, and then just related to that, do the enlarged ones, do you ever see any multivesiculation of them?
Yeah, we haven't looked, we
Okay.
don't have the resolution to look yet, but that's a great suggestion. Yeah.
I have a comment. Let me pull it up here. I looked through some data, and SORA1 expression is lowered in senescent human astrocytes as well, which are more abundant in AD.
Oh, that's interesting. Yeah.
Well, there are no other questions from the chat,
Great.
But thank you so much for your talk. All the speakers, or Dr. Young and Dr. Singhvi, will be available during the during the coffee break if you have any other further questions for them. And we'll reconvene at 2:20.
Yeah, I'm sure yeah, so we did a lot of, yeah.
Hello, hello. Can everyone hear me? No? Testing, testing. Can everyone hear me? Perfect. Awesome. So I hope you all had a nice coffee break and are recharged and ready for round 2 of metabolism and brain aging. Now we have Heidi Pack presenting her talk entitled Time of Day Dependent Response to Insulin Signaling in Calorie Restricted Mice.
Hello, I'm excited to present some of my data today, but I would like to first thank the organizers of the symposium for inviting me to share with you some of this exciting data I've been working on the last few years. And today I'm going to be talking about, my title's a little bit misleading, but today I'm going to be talking about time of feeding dependent response in calorie restricted mice. So there are a lot of interventions that delay the onset of aging, and a gold standard intervention that extends health and lifespan is calorie restriction. Calorie restriction is the reduction of energy intake without malnutrition, and it has been shown in various model organisms to extend health and lifespan. While this is simple in concept, it's very difficult for the average person to achieve calorie restriction. So, calorie restriction models have been done in various model organisms, and one of which is rhesus monkeys. And here I took this image from the New York Times, and they published this story from the rhesus monkey study on calorie restriction at the University of Wisconsin-Madison. Here on the left, you can see Kanto. He's on calorie restriction. He has a nice shiny coat with upright posture compared to his non-CR counterpart Owen, who has a slouch posture and wrinkled face. And below is a human equivalent diet, which they represent as a low energy dense food here in 3 meals, with the average food for a normal person in America with a high energy dense food represented here. But this is a little bit misleading, and I'm going to tell you why. So there's a key overlooked feature in calorie restriction, and I'm going to be talking about this in the context of murine models, as this is the model I use for my studies. For a typical ad libitum mouse, you give them unlimited access to food, and on a regular chow diet, they'll eat majority of their daily caloric intake during the dark cycle and little snacking during the light cycle. For calorie restricted animals, they develop a hyperphagic response in that they eat all their daily portion within about 2 hours. That means they are essentially fasting for 22 hours a day. And so this really became a question of, are calories really driving this response in calorie restriction, or is fasting required to drive the calorie restriction response? So I developed this multiple feeding paradigm to really address this question of calories versus fasting. Here you have the AL group, which is the standard control diet group on AdLib, and then the classical CR model is labeled as CR with 30% restriction and fed once a day. And you have the diluted AdLib group, which is a calorically restricted animal without fasting. So you lose the fasting aspect by getting rid of 50% of their bulk replacing it with indigestible cellulose. And then you have the MF.CR group, which is another calorically restricted animal. And then you eliminate the binging behavior of CR mice by feeding them 3 even portions during the dark cycle. And to really address the fasting question to really address the fasting aspect of calorie restriction, you needed to have a group that fasted as long as a CR animal, but ate almost a similar amount as an AdLib group. And here it's labeled as TR.AL. So this work is going to be published in Nature Metabolism soon. So I'm just going to summarize the findings of what I found in this study. And is that fasting is required to see this improved insulin sensitivity in CR mice. Fasting is required for the increase in fatty acid utilization in CR mice. And fasting is required for the distinct metabolomic signature you see in CR mice. And restriction of calories itself without fasting does not improve frailty or extend lifespan. And finally, fasting alone can recapitulate the CR effect and produce similar transcriptional landscape. So hopefully I have convinced you that CR mice are a fasting model. So this becomes really a question of what are you really studying when you compare a chronic fasting model versus a non-fasting model. And so what I wanted to do was characterize the physiological and molecular response to fasting in the AdLib and CR mice. And to achieve this, I tested their response to these tests every 4 hours during a 24-hour cycle. And these were done on B6 male mice. And I had 6 different cohorts for each time point, making the assumption that these are all genetically similar mice, so they should be essentially identical. And what I found that was that the CR mice had improved glucose tolerance at majority of the time points. Actually, they're essentially the same at all different time points. The AdLib group, as you prolong the fast, it looks like they have improved glucose tolerance towards the later fasting time points. But more interestingly, this is a surprising finding, is that they look like they were insulin resistant at the earlier fasting time points, so before 16 hours, and only showed insulin sensitivity after a 20-hour fast. And I also did this in aged mice. These are 19-month-old mice, and they show, the CR mice show similar trend in glucose tolerance or retain a similar trend in glucose tolerance as they age. Similar with insulin tolerance, they show similar trend as well, whereas the aged AdLib mice appear to be slightly insulin resistant at this concentration. I also assessed gluconeogenesis in these mice with alanine tolerance tests. So alanine tolerance tests, it is essentially like a glucose tolerance test, but you inject with alanine and see how much glucose is produced from this alanine. And here you can see that CR mice actually maintain their tight blood glucose homeostasis up to a 20-hour fast. So they're essentially not producing glucose. Whereas as you prolong the fast, the AdLib group produce more and more glucose over time. And so to really investigate this unique response in insulin in CR mice, I collected or measured their fasting insulin levels, fed them, and measured their fed insulin levels, and found very unusual levels of insulin in both AdLib and CR mice. But one thing I want to note is that the CR mice maintained a very tight blood glucose homeostasis, or tight blood glucose levels, up to a 20-hour fast, and by 24 hours they kind of lost this maintenance of their glucose level. And I kind of saw a trend in this data in that if you shift the data by 12 hours, so instead of 6 a.m., I shifted all the data to 6 p.m. of feeding of the AdLib mouse, the insulin levels align exactly to that of both AdLib and CR mice. So that was just a hypothesis at the point because I just shifted the data. So to really address this question, I did a second set of experiment where I shifted the timing of the feeding of the CR mice from 6 to 7 a.m. to 6 to 7 p.m., did the same exact type of study, and found there's no real change in glucose tolerance, except for that possibly the AdLib mice are actually worse off at the 24-hour fast. And for insulin tolerance, they retain the same trend. So this is where I say my title is a little bit misleading. So it's not time of day dependent response, but more of when the first meal was initiated. But just as predicted, the insulin level was exactly aligned when you shifted their feeding schedule. So this insulin secretion, and/or clearance is heavily dependent on time of the first meal. But this doesn't really answer the odd data that the insulin resistant question that I originally had in the why are they insulin resistant but maintain a tight blood glucose homeostasis. So here are some of the clues that I want to just summarize before we go on. And it's that CR mice maintain the same blood glucose tolerance at all time points. They also maintain tight blood glucose levels even after a meal. And they appear to suppress glucose production during a fast. And they appear to be insulin resistant before a 16-hour fast, as assessed by an insulin tolerance test. And insulin secretion and/or clearance appear to be regulated by the time of first meal. So based on these clues, I propose two hypotheses. The first one is insulin sensitivity may be the result of negative feedback regulation of mTORC1 on insulin action. And hypothesis 2 was that CR mice maintain tight blood glucose level via an insulin-independent mechanism. I decided to tackle hypothesis 1 first because it was a little bit more direct. And so we had a lovely introduction on mTOR earlier this morning, and it is a master regulator of many metabolic processes. And so it is also a regulator of insulin sensitivity via S6K1 feedback inhibition on the insulin receptor substrate. What I found was that mTOR activity was essentially the same for CR and ad-lib mice, except for the earlier fasting time point. So phosphorylation of S6K and S6 is a downstream readout of mTOR complex 1 activity. And here you can see that for CR mice, they have a hyperphosphorylation of S6 at the 4-hour fasting point, but by 8 hours, ad-lib mice and CR mice are essentially the same. Surprisingly, I didn't see a difference in phosphorylation of S6K. And unfortunately, I don't have the 16-hour time point in this figure, but more or less it is the same in the muscle as well, except for that phosphorylation of S6K was hyperphosphorylated at the 4-hour fasting point, and it looks like it's being reduced by the later fasting time point. And you see the same trend for phosphorylation of S6. So at least for phosphorylation of S6K, it might indicate why we see this insulin resistance when you test it with an insulin tolerance test, because an insulin tolerance test is slightly biased towards a muscle phenotype. So I kind of addressed the mTOR question, but now I wanted to tackle hypothesis 2, which that CR mice might maintain tight blood glucose level via an insulin-independent mechanism. And in order to answer this, I used diazoxide, which is a common drug to treat for hypoglycemia and hyperinsulinemia. And this drug inhibits secretion of insulin in the pancreas by opening potassium channels in the beta cell membrane. And to do these, what I was hoping for to do theoretically was to treat them with diazoxide, collect their fasting blood, feed them for about 2 hours, and then collect their fed blood once again. However, what I found out was when you treat mice with diazoxide, they don't want to eat right afterwards. More surprisingly, CR mice treated with diazoxide produce exorbitant amount of glucose compared to an ad-lib mouse treated with diazoxide. This suggests to us that perhaps CR mice suppress their gluconeogenesis during a fast, or they have a larger glycogen storage. And so just to summarize everything, the CR mice maintain same blood glucose tolerance at all time points. They have a very tight regulation of blood glucose, and they suppress glucose production during a fast. And they seem to be insulin resistant before a 16-hour fast, as assessed by an insulin tolerance test. And insulin secretion and/or clearance appear to be regulated by the time of first meal. And lastly, insulin is required for the tight regulation of glucose homeostasis in CR mice. So because this was a short talk, I wasn't able to present some of my other data. Currently, I'm working on this, really looking into the suppression of gluconeogenesis, or if they have increased glycogen content. I didn't show any of the data where what mTOR activity looked in the fed conditions. I also looked at other mTOR targets as well. I didn't really see a difference, but I didn't show that. I'm also doing a secondary verification of mTOR activity in the liver by using a TSC1 liver knockout mice, which is a hyperactive mTOR model. And I'm looking at other nutrient responsive pathways outside of mTOR and mediating the CR response. And so I would just like to thank everyone in my lab, particularly for my 5 different feeding paradigms. Cara, Nicole Richardson, Sabrina, and our collaborators, Spencer Hawes and Mitchell Lavarius, really helped out with the work. And for my current 24-hour study, Cara was a great deal of help, as well as my undergraduates.
Wonderful talk. Thank you so much. I was interested in whether you think that if you shifted the circadian rhythm of the mice artificially, would you still see sort of similar patterns at different times of day, or do you think it sort of has some relativity to what their particular circadian rhythm is?
So that is a great question. My data suggests that this is not dependent on light and dark cycle. It's mostly a timing of their food intake. And there's research out there where they have done feeding rhythms versus light and dark cycle. So the light and dark cycle will dictate their behavior to feeding. But this can be overridden, like in a lab setting, when you restrict them to a certain time of feeding. So that's what's happening with these CR mice.
Interesting. Thank you.
We do have an online question, and feel free, online participants, to write in the chat. And if for some reason the chat's not working, feel free to contact me or any of the panelists directly. This is from Alessandro. He says, great talk, Heidi. Considering that the S6K1 feedback is thought to be the cause of so-called rapamycin-induced insulin resistance, would it be possible to see similar changes in insulin sensitivity in response to a single bout of rapamycin?
That is a great question. I did test this out before. They actually appear, when you do a single bout of rapamycin, they appear insulin resistant at the time point that I use. So I chose a 4-hour fasting time point because that's where they looked like they were insulin resistant. So I thought perhaps by injecting them with rapamycin, it might rescue that insulin resistance. I actually saw that they seemed even more insulin resistant compared to their vehicle-treated mouse.
Thank you so much for your talk.
Thank you.
Next up, we have Elizabeth Ray, who will be presenting her talk entitled CNS Insulin in Alzheimer's Disease.
Thank you, Rachel. And thank you to Alessandro and Mitchell for the invitation to present to y'all today in person. And thank you to Rose and Shelby for organizing the behind the scenes aspect of the symposium. I don't know why this new tab. Oops. All right, so I'm a research assistant professor in the Division of Gerontology and Geriatric Medicine at UW. My lab is based down at the VA Puget Sound, but we have many overlapping works with some of the UW labs. And so today I'm not only going to introduce you to some of my personal research interests in CNS insulin and Alzheimer's disease, but as a product of the Genetic Approaches to Aging Training Grant from 2014 to 2017, I hope to also introduce some I guess, useful tools for the trainees in the audience, both here and online, to try to take advantage of while you are in that setting, because you'll never have another experience like it. So as some of the other presenters have already introduced today, which is nice, I want to first start off by telling you a little bit about central nervous system insulin and how it acts. And so as we just heard from Heidi, it has this huge metabolic role within the brain. And so it can act as an anorexigenic hormone, decreasing food intake. It also has these effects on peripheral hepatic glucose production while acting within the central nervous system. And it can really act on particular cell types, such as POMC neurons, to regulate peripheral metabolism as well. But my main research interests are in its cognitive role, in which it can be neuroprotective, promote dendritic sprouting, as Dr. Sell presented earlier, can be important for cell growth, and my main interests are in its role for improving memory. And so in the last couple of decades, there's been this strong research performed on the degree of CNS insulin resistance in Alzheimer's disease. And this has arisen due to various factors that people have shown throughout the literature. First is that there is a decreased cerebrospinal fluid to serum ratio for insulin in people with MCI as well as Alzheimer's disease. Additionally, there is impaired insulin signaling in postmortem Alzheimer's disease brain tissue. Related to aging, there is a decreased insulin receptor density present within the brain. And then lastly, there are diseases associated with deficient insulin action, such as diabetes and obesity, are also risk factors for the development of cognitive impairment. And so part of my postdoc work when I came to Seattle, to the University of Washington, was to look at the impact of insulin delivered intranasally and its impact on hippocampal gene regulation, as there had been previous studies shown that this type of therapy, intranasal insulin delivery, could improve cognition in both patients with mild cognitive impairment as well as Alzheimer's disease. So as I mentioned, when I came to Seattle, I was a part of the Genetic Approaches to Aging training grant. And so, sorry for the cutoff here, but being a part of this training grant allowed me to attend some of the spring seminar series that the training grant holds every year. And Holly Brownborg was an investigator that came to one of those seminars. And I was able to have lunch with her. And she introduced me to this Neurobiology and Neuroendocrinology of Aging International Symposium that she organizes every other year in Bregenz, Austria. And so it's held in the even numbered years. So the next one is hopefully in July of 2022. And she's designed this so that it coincides with the opera festival. So here on the lower right, you can see this beautiful opera stage on the water. And the operas rotate every 2 years. So every time an attendee comes back, they get to see this new opera. But the social aspect wasn't the most exciting part of the symposium. It was also the science that was presented that was so relevant to what I was doing with intranasal insulin and gene regulation in the hippocampus. And so as part of this meeting, she also puts together a special issue in experimental gerontology, in which she invites some of the attendees to submit their own works for publication. So I was able to review the literature in my field at that time and get it published shortly thereafter. So some of the work that I presented at that meeting was looking at gene regulation, as I mentioned. And so here I took young and aged SAMP8 mice, which as mentioned before is an accelerated aging line. And I also took aged SAMP8 mice that received an acute intranasal insulin injection. And so you can see the genes that are either downregulated or upregulated in each of these cohorts of mice. And so the aged vehicle mice have this robust change with gene regulation in the hippocampus. And when we treat with insulin, we get this slight restoration back to that young phenotype. In combination with these studies, I also wanted to look at transport of insulin following intranasal delivery. So where does insulin go in the brain when we deliver it? And so looking in the whole brain, you can see in the black lines here,
we get this nice distribution throughout the brain and uptake over time in a matter of 1 hour. However, I wanted to investigate one of the mechanisms for this transport. And so it was thought at the time that the insulin receptor played a role in transporting insulin throughout the brain into various tissues. This was the field. So I used a selective inhibitor for the insulin receptor S961 and found that this completely blocked intranasal insulin transport. So this was in line with what the field was thinking. But this wasn't enough for me because this was just intranasal delivery. This was an exogenous delivery of insulin. I wanted to look at the endogenous processes. So I was able to apply for this travel fellowship through the Company of Biologists and take advantage to going to a collaborators research group in Melbourne, Australia, to look at the transport of insulin across an ex vivo epithelium. And so these are porcine cheek epithelium slices that you can put in a dish and measure the amount of insulin transport across, or any type of substrate. And so this was to represent more of that intranasal process, the intranasal epithelium, and see if we could modify insulin transport. But part of this coincided with the meeting, the cerebrovascular biology meeting that's held every other year. And in this case, it was held in Melbourne. And so I was able to present some work related directly to the blood-brain barrier, which is another research interest of mine. So as Kira already introduced earlier, this blood-brain barrier is a network of cells. You have endothelial cells that are tightly connected by tight junctions, and these are surrounded by pericytes and astrocytic infeed that completely ensheath these brain endothelial cells. So these cells are really the gatekeepers of the brain, and they help to regulate what gets in and what gets out. This is the bane of pharmaceutical companies in trying to get drugs into the brain, because these cells are so tightly controlled that they can just push these drugs back out of the brain without them having an effect. So I wanted to look at the endogenous insulin transport system. And so to do this, we can radioactively label a peptide, in this case insulin, inject it intravenously in mice and allow it to circulate, and collecting blood and brains from different mice throughout the time course, we can then measure radioactivity in these samples. And so when we plot the brain serum ratio of radioactivity over time, we can get a slope of transport, which is indicative of the rate of transport, or the Ki. But what's also important from these studies is that we also get the level of reversible binding to the endothelium, which is the Y-intercept, or VI. And so here in the same study, where I gave that same inhibitor to the insulin receptor, which should block insulin transport, because that's what the field believed at the time, and that's what happened in my intranasal studies, we still see transport of insulin across the blood-brain barrier. And so what we did find is, you'll notice this extreme decrease in the VI, or the level of reversible binding. And this makes sense because we have eliminated that primary source of insulin binding on the endothelial cell surface, the insulin receptor. So insulin can't bind, so we're not seeing that. I repeated these studies in a genetic model of endothelial insulin receptor knockout mice and saw the same exact thing. So it seems to hold true that following intranasal delivery in epithelial cells, the insulin receptor is responsible for transport into the brain, whereas across the blood-brain barrier, this endogenous transport system, the insulin receptor may not be involved in transport. So after generating this data, this was towards the end of my postdoc fellowship, and the training grant also introduced me to the Butler Williams Scholars Program, which is unique to aging investigators. And so it's a chance for young trainees like myself to travel to Bethesda, Maryland, and actually go to NIH and sit in a group with a lot of other young aging trainees and work through grant ideas and specific aims page. And so this was my first real formal introduction to writing grants. And it was a great opportunity to experience it and see how study sections are run and what they look for and how they grade these grants that we would be submitting in the future. So many years later, I was fortunate to get a supplement that I just recently received to conduct some further studies investigating insulin transport at the blood-brain barrier. And this involves the whole neurovascular unit as a whole. So we also got an introduction from a couple of the earlier speakers today about this communication between these cells within the CNS. So this is a busy slide, but it highlights all of, not all, but a few of the signaling capabilities between astrocytes and brain endothelial cells, between neurons and astrocytes, and this communication network in all of these cell types. And so I am primarily interested in what does the role of the insulin receptor on astrocytes do in regulating brain endothelial cell transport or blood-brain barrier transport. So if we knock this insulin receptor out, I was able to receive some of these genetically modified mice where the mice lack the insulin receptor on astrocytes from a group at Joslin Diabetes Center. And I was able to repeat those exact same pharmacokinetic studies to look at blood-brain barrier transport. And what I hope you can appreciate is that this slope of this line is nearly flattened compared to the wild type controls. And so it's reduced to about 25% of that level of wild type mice in terms of transport. So this data suggests that the astrocyte insulin receptor may actually be involved in regulating blood-brain barrier transport. And so the supplement that I just received is designed to further investigate why this might be going on and how these cells might be communicating. So through this process from postdoc to now my research assistant professor position, I've been fortunate to really dive into the field of insulin interactions at the blood-brain barrier. And I was fortunate to publish this recent historical perspective on these interactions at the blood-brain barrier last year with my colleague and co-author Bill Banks, who was my postdoc mentor, on how the field has evolved over the last 100 years since insulin was even discovered. So last year we celebrated the 100 years since the discovery of insulin, or I guess this year, excuse me. And so we were able to kind of take it decade by decade how this field progressed. And I've been fortunate to contribute to this field in looking at various modulators and interventions of insulin blood-brain barrier transport, including apolipoprotein E, sex, and high-fat diet, all of which we've published. Rapamycin and rosiglitazone, I recently published those effects on insulin blood-brain barrier transport. And my most recent projects involve looking at the roles of the soluble form of the insulin receptor and whether this mediates insulin blood-brain barrier transport, as well as exercise. Lastly, I've also looked into endocytosis processes, trying to define the differences between the insulin receptor and the insulin transporter at the blood-brain barrier, and now this neurovascular unit contribution. So with that, I'd like to thank my lab members, most of which are current and previous lab members, most of which are undergrads or med students that have helped throughout this project. The Banks and Erickson labs at the VA as well that have helped provide both technical and intellectual advice. My collaborators that helped generate some of this data, my funding sources, and as a junior investigator, I also have a QR code to a survey link if you don't mind scanning it with your phone to just help provide feedback for future presentations and talks. I'd really appreciate that. Thank you so much, and I'll be happy to take questions.
Okay, fabulous talk. Are there any questions?
Very nice
Thank
talk.
you.
Thank you very much. I was thinking about the insulin receptor isoforms. So they vary in the brain. And now you have forgotten exactly the splicing or whatever. Right? Now the A and B form. Does it vary?
That's a good question. So there are still the A and B forms. One is more predominant than the other. Which one it is, I will have to get back to you on that.
In the IGF-1 receptor?
That I'm not sure. I haven't really explored the IGF-1 field. Thanks. Oh yeah. So Dr. Sell asked about the IGF-1 receptor and whether or not it was the same in the brain versus the periphery, or if it's involved in transport. Yeah. So previous studies have looked at the role of IGF-1 and its effects on blood-brain barrier transport. for insulin, and they've shown no real overlap except in extreme conditions where you really oversaturate one process versus the other. Thanks.
Thank you very much.
We have a comment from Alessandro who says, "Thank you, Elizabeth, for highlighting the great opportunities that stemmed from your association with the training grant and the Nathan Schock Center." And with that, thank you very much for your talk.
Thank you.
Right, next up, we have a virtual presenter. Sarah Acanyas is going to present her talk entitled "Sex Differences in Microglial Transcriptomic Responses to Aging and Alzheimer's Disease Pathology."
Can everyone hear me and see my slides? Okay, good. Thank you. Thank you for the introduction. My name's Sarah Acanyas, and I'm a fifth-year PhD candidate in the Department of Physiology at the University of Oklahoma Health Science Center, and my mentor is Dr. Bill Freeman. So I'd like to thank the organizers for inviting me to talk today about sex differences in the microglial transcriptomic response to aging and AD pathology. So as many people have talked about today, Alzheimer's disease is the most common form of dementia in older adults. And it's characterized by a decline in memory, reasoning, and other thinking skills. Some of the pathological hallmarks of AD include positive lesions in the brain, including amyloid plaques, hyperphosphorylated tau, and neurofibrillary tangles in the neurons. And really central to all of this is microglial cell activation. So the microglial respond to the proteinopathies, and they phagocytose parts of neurons. They release pro-inflammatory cytokines and chemokines, and may contribute to the negative lesions or neuronal and synaptic loss that really contribute to the cognitive decline. So there are well-documented sex differences in Alzheimer's disease. Females have a higher prevalence of AD, and it's almost a 2-to-1 ratio. Females also have worse pathology, as indicated by post-mortem brain analysis. Females have more severe cognitive decline, so they perform worse on cognitive tests. However, males with AD tend to progress to death more quickly. So it's clear that there is some basic biological differences between males and females that make them respond to AD pathology differently. So central to my research is the question, what mechanisms mediate sex differences in the prevalence and progression of AD? So recent GWAS studies have shown that the majority of the late onset Alzheimer's disease risk alleles are in innate immune system related genes, and they're largely microglial genes. One of the most popular of these is TREM2, which is a microglial gene, and you see here. So this figure over here is from a paper in Brain Communications, where they show that the amyloid, in an amyloid mouse model, the APP-PS1 mouse model, they did hippocampal gene expression, and they show this co-expression network of microglial genes are mostly these AD risk loci. And so this has really shifted the field to focus on potential causative roles of microglia in AD pathogenesis. So even in a healthy brain, microglia have this shift from a homeostatic surveillance phenotype into this primed inflammatory phenotype where they increase in proliferation and are expressing pro-inflammatory cytokines and chemokines and phagocytosing different things within the brain milieu. And so our question is, what causes this shift in phenotype? And are there sex differences in this shift to a primed state? So a previous study out of our lab has shown from whole tissue hippocampal transcriptomics that there's a sexual divergence of neuroinflammatory pathways with brain aging. And we say sex divergence because we only see these differences when we compare old males and females, and they're absent in young and adults. And if you noticed, most of these pathways that are different in the hippocampus between young, old males and females are primarily microglial pathways like complement, inflammatory response, macrophage activation, and antigen presenting cell activation. And so we know that there's sex differences in AD, and we know that microglia become activated with aging and can contribute to disease. And so it's possible that sex differences in microglia are mediating the sexually divergent response to AD. However, there's a knowledge gap right now as to the microglial contribution to sex effects in the brain with aging and AD pathology. So we performed an experiment on C57 black 6 mice where we took male and female, young and old mice, and we dissected the hippocampus and created a single cell suspension. A recent concern with just the creation of a single cell suspension from brain tissue is that it can result in ex vivo activation, which may mask differences between your experimental groups. So we recently submitted a manuscript where we performed an experiment to compare ribotag or ribosomal tagging approach, TRAP, to traditional self-sorting methodologies to see what the contribution of creating a cell suspension was in inducing ex vivo artifacts. And so just briefly, what we saw was indeed the creation of a cell suspension dramatically induced ex vivo activational artifacts, and that the inclusion of transcription and translation inhibitors could prevent these changes. So we are working on reviews that we got back from eNeuro, and this manuscript is available on bioRxiv if you're interested. So for this experiment, we included these transcription and translation inhibitors to prevent the ex vivo activation. We performed magnetic activated cell sorting to capture CD11b positive cells from the hippocampus. And we assessed the purity by flow cytometry. And so you can see when you compare our input to positive fraction, we have good enrichment of CD11b, CD45 double positive cells, around 80%, with no difference between our experimental groups. One concern is that we may also have, since we're capturing CD11b positive cells, we may also capture circulating monocytes or other brain resident macrophages. Microglia are usually identified as being CD45 mid, whereas circulating monocytes and other macrophages have high expression of CD45. So you can see here our cell population is mostly CD45 mid. So we believe that we're capturing the majority of microglia. We then took our CD11b positive cells from the hippocampus and performed RNA sequencing. And our principal component analysis shows clear separation of young and old. So there's a great age effect in microglia. And what you can see here is the young males and females cluster together, whereas it looks like the old males and females are starting to diverge. We called differentially expressed genes with a main effect of age. And we had around 3,700 genes that were differentially expressed in microglia with age. And the majority, 2,000 of them, were differentially expressed both in males and females with aging, although females had a much higher number of differentially expressed genes than did males. And you can see the genes that were differentially expressed by age. You have a subset that are upregulated with age, and you have a subset that are downregulated with age. To assess the biological significance of this, we did enrichment analysis, Go biological process analysis. And we saw several pathways of interest, including, again, the antigen processing and presentation, and also cell chemotaxis. And if we look at some of the individual genes involved in antigen presentation, we see some genes that are sex common changes. So they're changing both, the red is males and then the blue is females. They're changing both in males and females with aging. And it seems to be about the same magnitude of change in both males and females. But then we also have a few genes that have a female bias change in expression with aging, including CTSE and SLC11A1. So this process of antigen presentation may be different between males and females at an old age. When we look at KEGG pathways, so another pathway analysis, we see again our antigen processing and presentation, and then also some disease pathways and lysosome and phagosome pathways. So I was interested to see with the Alzheimer's disease pathways if some of those risk alleles were different between males and females. And so I looked at TREM2, ApoE, PTK2B, and ABI3, which are all known to be associated with AD. And we see female-specific changes with age in the microglial expression of these genes with no significant change in the males. So I found this to be very interesting that the genes involved in Alzheimer's disease may be changing differently by sex. And then in addition, there's some of those AD risk loci genes that are changing in both sexes to the same degree. We then looked at the genes that were differentially expressed with a main effect of sex, and we found 186 genes. And not surprisingly, we had more differentially expressed genes by sex in our old animals than in the young. And they're separating here in the first component by sex. So we have our males versus our females. And then in the second component by age. When we look at Go biological processes that are enriched in this set of genes, we see again a lot of inflammatory pathways, including the positive regulation of cytokine production. And when we look at this pathway on a gene level, we see larger differences when we compare old males to old females than in the young group. So there seems to be, again, this divergence in phenotype in old animals. So after completing that study, we were interested in how that would compare to AD pathology. So Dr. Beckstead at OMRF gave us some APP-PS1 mice, which is an amyloid model of AD. And we performed a similar experiment, except instead of doing aging, we compared AD mice to controls and then did RNA sequencing. Very similar to what we saw with aging, we had a high number of genes, around 5,000, that were differentially expressed by AD pathology, so AD versus control. And most of these were in common between males and females, although again, females had much higher number of genes that were differentially expressed than did males. And again, we have a subset of those genes which are downregulated in AD and a subset of the genes that are upregulated in AD. We then looked at our pathways, KEGG pathways, and we saw, again, our disease pathways for Alzheimer's disease and Parkinson's disease as enriched, as well as this lysosome pathway, which are all very similar to what we saw in our aging study. However, when we look at the expression of these AD risk loci genes, we didn't see the sex differences that we saw with aging. And this may be because this experiment was done in adult mice instead of aged mice. And so we see sex-independent changes in the gene expression of these risk loci genes. There were a few of the AD risk loci genes that were sex-dependent. And there's one that was female biased, and then 2 of them that were male biased, which is different than what we saw with aging. We then wanted to compare our aging study to our AD experiment. And so we took those differentially expressed gene lists and did a 4-way Venn diagram. And then to prioritize the intersections, we converted this Venn diagram into an upset plot. So each of these bars is just those intersections ranked by the number of genes in common. And then down here we have our 4 comparisons. So the highest number of comparisons were our AD specific changes that weren't seen with aging. So AD versus control, both in females and males. And when we did pathway analysis on these 1,600 genes, we saw the pathways for N-glycan biosynthesis and also fatty acid metabolism. And when we look at this on the gene level, we see very clear separation of our control and AD groups with really no effect of sex here for both of these pathways. The next intersection we looked at was common between all of our groups, so AD and age changes that were sex independent. And when we ran pathway analysis, we saw pathways for Alzheimer's disease, Parkinson's, Huntington's, lysosome, and oxidative phosphorylation, which I've already showed in the previous slides. And then our next intersection here is, these are age effects, so old to young, both in males and females, sex independent. And here we see pathways for antigen processing and presentation, as well as autoimmune. Here we had 146 genes that were common between AD and age, but female specific. But when we ran pathway analysis, we did not get any significant pathways from those 146 genes. And here at the very end, these 4 in common were male-specific changes by AD and age. And so it seems that microglia have many sex-common and sex-divergent transcriptomic responses to aging and AD. And many of the changes that we see with aging are the same changes that occur in response to AD pathology. However, there are some important differences. Those age-specific changes were in autoimmune pathways and also antigen processing and presentation, whereas the AD-specific changes were almost exclusively in metabolic pathways. And our age and AD common changes were enriched in disease pathways like Parkinson's, Huntington's, and AD. So our future directions, where we're going next, we're going to compare this study to an ongoing aging study that we have in the lab using ribosomal and nuclear tagging NuTrap approach to isolate microglial nucleic acids, so DNA and RNA, without the need for cell sorting. And the benefit of this is we can lineage trace the same cell population to really look at how that cell population is changing with aging, because markers like CD11b could potentially change with age or disease. We're also working on epigenomics, we ran whole genome oxidative bisulfite sequencing to look at methylation and hydroxymethylation. We're attempting to disentangle the relative contributions of gonadal and chromosomal sex in regulating hippocampal sex differences to look more mechanistically. And we have recently submitted a manuscript to molecular neurobiology. It's also on bioRxiv using the 4-core genotype model. And we're planning to cross the 4-core genotype model with an AD mouse model so we can look at relative contributions of gonadal and chromosomal sex as well. So with that, I'd like to thank my lab, my mentor, Dr. Freeman, and also Dr. Anachuker Elliott, who has helped me optimize a lot of these techniques. Kevin Pham, who did a lot of the experiments with me and helped produce a lot of the figures. Mike Beckstead, Dr. Beckstead, he gave us those APPPS1 mice, all of our core facilities, our funding, and then the UW Nathan Schock Center for inviting me today. And with that, I'll take any questions.
All right, thank you so much for your talk. We do have a couple online questions. The first one is from Sylvan Urfer. Apologies if I mispronounced that. There is some evidence that taller people are somewhat less susceptible to Alzheimer's disease. How much of the sex difference you observe in humans could be explained by that mechanism?
So I don't know if I'm biased, but I think it might be the other way around, in the sense that maybe the differences in the height could be explained by sex differences. I don't know what mechanism between short and tall people that would make a difference, but I haven't heard that. I'll have to research more into it.
The second question is from an anonymous attendee. The separation of gonadal and chromosomal sex seems important since mice do not experience menopause. Can you briefly highlight the limitations in translating these findings to humans?
So I would agree. That's actually where I want to go in the future with studies, is to use the 4-core genotype model on gonadectomized mice to see the differences between males and females. So there's definitely limitations in any model, but I still think that the differences that we're seeing here can be translated to humans, because we see a similar trend in humans as well.
Well, thank you very much for your talk.
Thank you.
So lastly, just to close off this conference, I wanted to give one last round of applause to all the speakers who presented today. It was an incredibly fantastic talk. I want to give another thank you to Mitchell and Alessandro, who were the fabulous organizers behind the symposium. And also a thank you to Rose and Shelby for coordinating all the behind the scenes efforts. And thank you all for coming today. Following this is a BMHA retreat. All the speakers are invited. We highly encourage you to attend. That will be at 4 p.m. at Republic. And Rose has some information about parking validation.
Okay, so I'm really short. If you guys did park in the parking garage, please validate your parking with the front desk before you go to Republic or before you leave, because their desk does close at 5:30. We are going to have access to the parking garage after the fact. We have a security guard that'll be waiting to let us in so we can get access to the parking garage. Yeah. So it's basically how like any parking garage works, where like how you came in, you got the ticket, there was another gate on the other side where you would have to be able to scan that to get out. Yeah, but just make sure that you validate it with the front desk, because they should give you like that separate ticket.
All right, thank you.